At the end of the previous video, we learned about secondary active sites that are located away from the main active sites, which are what we call allosteric sites, meaning other sites or secondary active sites. Now, what this means is that a different substrate with a different size and different chemical properties is actually able to enter the allosteric site, even though the substrate that enters the main active site is a completely different size with completely different chemical properties. And these allosteric substrates broadly are what we call allosteric effectors, but we're going to give them more specific names based on the effects that they have on the enzyme. So this particular allosteric effector is what we call an allosteric activator because of the effect that it has on the active site. So before the effector has bound, we can see that the shape of this active site is a very poor match for this substrate, which makes it difficult, but theoretically not impossible because of induced fit, to enter this active site here. But when the effector, specifically the activator, binds to the enzyme via the allosteric site, the conformational changes that it causes to the enzyme result in the active site opening up and becoming more suitable or more complementary to allow the proper substrate into the active site, which in general makes it much easier for the substrate to enter the active site and for the enzyme to begin catalysis. Because this is all caused by an allosteric activator, we call this process, not surprisingly, allosteric activation. Now, the opposite process of allosteric activation is what we call allosteric inhibition. So if you've never seen the word inhibition before, the verb form of this word is the verb to inhibit, which is a different way of saying to prevent, which means that somehow our allosteric effector is going to prevent the enzyme from functioning in its normal way. So the allosteric effector in this case is what we call an inhibitor rather than an allosteric activator. And again, it functions more or less in the same way, but with the opposite consequences as an allosteric activator, in that it binds to an enzyme via the allosteric site, which we can see here, but instead of causing the active site to open and become more accessible to the substrate, it actually causes the active site to close and therefore become less accessible for the substrate. So here we can see that the conformational change in the enzyme because of the inhibitor has closed off the active site, which means that it is no longer possible for the substrate to enter the active site, and therefore this enzyme is effectively rendered off or inactive. And that's actually the primary way that inhibitors and activators function in the human body. Most allosteric activators or allosteric inhibitors are actually naturally occurring compounds in the body, and depending on whether you have more activators or more inhibitors, it increases or changes the likelihood of whether the enzyme will be on or active, or whether the enzyme will be off. And this is a good way for the body to regulate its own catalysts without having to constantly break them down and make more of them. Instead, it can simply turn them off by adding an inhibitor or turn them on by adding an activator. Now, because an allosteric substrate does not actually enter the primary active site, meaning that it doesn't compete directly with the substrate in order for bonding to the same location, allosteric inhibition is what we call non-competitive inhibition. So if non-competitive inhibition exists, one would assume that competitive inhibition exists, meaning that some inhibitors, instead of binding to an allosteric site and indirectly inactivating this enzyme, some inhibitors may actually compete with the same site as the substrate, and that's exactly what we see with the concept of competitive inhibition. So as I write this down, we can take a look at what is going on in terms of the inhibitor, which we see in green, and the substrate here, which I will label. So the idea is that a competitive inhibitor, rather than an allosteric inhibitor, binds directly to the active site of an enzyme. And when we say binds, this doesn't necessarily mean that it's creating a covalent bond. In fact, this particular inhibitor, let's say, for example, that it's polar with a number of noticeable positive poles in its structure here. Now, it can fit inside of the active site because if the active site has some complementary negative poles, it can sit inside of the active site and basically remain there because of the attraction of hydrogen bonds. And as long as this inhibitor is in the active site, this inhibitor prevents the substrate, regardless of whether the substrate matches or not. If this inhibitor is not here, the substrate is no longer able to get in. Now, in this case, we've said that the inhibitor is simply held in the active site by hydrogen bonds, and hydrogen bonds are not actually permanent interactions, because if this inhibitor were to be jostled, there's no covalent bond or ionic bond that is holding it permanently in place. So we say that this interaction with the inhibitor and the active site is only temporary, because, again, there is no permanent ionic or covalent bond that is holding it there, which technically means that this inhibition is actually reversible. So if we wanted to kick this inhibitor out of the active site, all that we would need to do is increase the amount of substrate, so our substrate concentration, so that we have more substrate molecules. And if we have a number of them that can compete with the inhibitor, you can actually get the substrate to either, number one, beat the inhibitor to the active site, or number two, actually physically kick out or displace the inhibitor and stop it from being able to block the active site. However, as we'll see in a number of cases, this action here, the displacement of the inhibitor, is not actually possible, because sometimes the inhibitor will actually form a permanent bond with the active site here. So in this case, it can be one of two things. Number one, the inhibitor can actually form a covalent bond with one of the R groups, or perhaps even the backbone of the active site, or of the protein that composes the active site. And another possibility is if it has a large number of positive charges, for example, and if the enzyme's active site has a large number of negative charges, it can form strong ionic bonds that will anchor the inhibitor in the active site and make it difficult to remove. When this happens, we can say that the inhibitor has actually permanently attached to the active site. Now, this is a very big problem, because this means that if the inhibitor binds irreversibly, meaning that we can't get it out, this inhibition is actually permanent. And if we don't have any way to actually break these bonds and get the inhibitor out, this enzyme has just been rendered useless. Now, technically, there are ways to break ionic and covalent bonds. For example, if there's a covalent bond that was formed by dehydration synthesis, you could do a hydrolysis reaction, but unfortunately, your cells are not really that efficient, and normally the only way to make this protein usable again is to break down the protein and remake additional enzymes that aren't blocked by this competitive and permanent, irreversible inhibitor. Now, as a consequence of how devastating this is, the fact that one inhibitor is all that's necessary in order to permanently break an enzyme, it shouldn't come as a surprise that many irreversible competitive inhibitors are actually poisons and some of the most deadly substances that exist on Earth. And we can look no farther than hemoglobin for one common example. Most people have an understanding that carbon monoxide is incredibly deadly because carbon monoxide, if we compare it to the structure of oxygen gas, has a similar shape. Both of them are diatomic, two atoms. Both of them have a straight linear shape, and both of them have electrons equally distributed on both sides, meaning that they also have the same polarity, given that they are both effectively nonpolar compounds, although yes, carbon monoxide does have a slight dipole because of electronegativity differences. But the fact that their polarity and shape are similar enough between each other, this means that carbon monoxide can actually bind to the oxygen binding sites that we see on hemoglobin, and when it does, this lone pair that is attached to carbon will actually permanently attach to the oxygen binding site and therefore render hemoglobin useless because it is no longer able to pick up oxygen, and there is no way to get a carbon monoxide molecule off, meaning that this hemoglobin molecule will have to be broken down and a new one will have to be made. And this is devastating for humans and anything else that requires oxygen, because if all of your hemoglobin becomes blocked by carbon monoxide, you have just lost your ability to transport oxygen, and therefore you have effectively killed yourself by a lack of oxygen. Now that we've finished learning about the different ways that you can regulate enzymes and how enzymes function, firstly, take a look at this case study involving one of the most famous enzymes in the human body and see if you can answer the questions about how pepsin, the enzyme found in the stomach, responds to its environment, concentrations of its substrate, and the different ways that pepsin is inhibited and regulated by the body.